All right, guys, 7.3 is going to look at how plants fix carbon in the carbon dioxide that they intake in the stomata or through the stomata, and how they fix that carbon dioxide into glucose. We have two products from the light reactions that are going to come in aid in the Calvin cycle. So those two products from the light reactions are ATP and NADPH. Remember, those are high energy molecules that are going to fuel the Calvin cycle. The Calvin cycle is going to take place in the stroma of the chloroplast, and there are just a series of enzymatic reactions that's going to take, again, carbon dioxide and produce a glucose at the end. We have three parts here. I don't have you write these down initially because we're going to go into each part here a little bit more in depth. But it goes carbon fixation, carbon reduction, and the regeneration of RuBP. Now, the Calvin cycle is very complicated. I want you to know basics about each of these three steps. I would also give you the hint of watch where the carbons are. There are complex structures here and just crazy amounts of atoms. I want you to understand where the carbons are. It's really important about the carbon. The Calvin cycle starts off with carbon dioxide fixation. This is when the Calvin cycle is going to intake a carbon dioxide molecule and attach it to something called RuBP. You can see RuBP down here has 5 carbons. Each one of these like purplish like circles represents a carbon molecule. Now, yes, it has 2 phosphate groups, but again, focus on the carbons. Understand that RuBP is a very complex molecule that has 5 carbons in it, but I just want you to know that it has 5 carbons. The Calvin cycle does this by an enzyme called Ribisco. Ribisco is the most abundant enzyme on Earth because it reacts very slowly, so there needs to be an abundant amount of it in plants, and there are a lot of plants, so there is going to be a lot of Ribisco on Earth. So when you take a carbon dioxide molecule and fix it to RuBP, it becomes this 6-carbon molecule. You don't need to know that name. The reason being is because it's very unstable and breaks down to 2-3 PGAs. Okay, so again, we had a 5-carbon molecule. We attached a carbon dioxide, which is another carbon. It made this 6-carbon molecule. Now there are 2 3-carbon molecules called 3-PGA. I love this diagram of this reaction. You can see here we have the RuBP, which is 5 atoms of carbon. We're going to attach this CO2 molecule. Now, this CO2 is going to actually be fused in the middle here. There's a misconception that it's added on the end. Nope, it actually is added in the middle here. And you can see where that carbon molecule here is on this 3-PGA, and then we have the other 3-PGA. Now, don't think that this carbon molecule is anything different than any of these other carbons. It's the same thing, and you'll see a little bit more in depth of why that is later on. Like I said before, Ribisco is this enzyme, most abundant enzyme on Earth, again, found in the stroma of the chloroplast. It takes CO2 and binds it with ribulose biphosphate. That's at RuBP. All right, our next step in the Calvin cycle is the carbon dioxide reduction. So what comes in here and is going to react with the 3-PGA is 6 molecules of ATP and 6 molecules of NADPH. We got those from what? We got those from the light reactions, okay? ATP and NADPH are high energy molecules, and they're going to energize, and they are going to give or at least change the 3-PGAs with the help of many enzymes into make something called G3P. Now, we're going to talk about the ratio of carbons here and as it transfers, what's going to happen here. But we're going to make this G3P molecule. G3P molecules are going to go on to make glucose. So this is where we're going to get our glucose at. Now, you should be kind of scratching your head because you only see 3 carbons here. And how many carbons are made of glucose? Yeah, you need 6 carbons to make glucose. In this case right here, we're going to need 2 G3Ps. So this whole cycle is going to have to turn twice for us to make 1 glucose molecule. If you do see something called PGAL when we're making diagrams and you're reading through this and trying to memorize this, PGAL and G3P are the same molecule. All right, this is the last part of the Calvin cycle, and this is where it gets difficult. One of those G3Ps is made into glucose. The other ones are going to be reformed into make RuBP. Now, you're probably going to scratch your head here and be like, we don't have enough carbons to do this. All right, so this is where in the beginning we had RuBP, which is 5 carbons long. It's going to bond with a CO2, which only has 1 carbon. It then made that 6-carbon molecule that broke down into 2 molecules of 3-PGA. Those 3 PGA molecules got turned into G3Ps. So what initially happens is you actually have 3 of those RuBPs that make 3 6-carbon molecules that make 6 molecules of 3 PGA that make 6 molecules of G3P. 5 of those are going to be recycled to make RuBP. Here's where you can do some easy math. 5 molecules of G3P times 3 carbon atoms is 15 carbon atoms. 3 RuBPs that are 5 carbon longs, 3 times 5, 15. We have enough carbon here. This is just a recycling cycle. It means it's going to just keep on continuously going on. But the main thing to understand here is you need ATP, which we're going to get from light reactions. But these carbons that are in G3P, where do they come from? They come from the CO2 you breathe in, or that plants breathe in, not you breathe in, that plants breathe in. This process is difficult. I would watch a couple videos, rewatch this. I did a pretty good job of not messing up the molecules. It's difficult. And what I'm going to tell you is, again, watch the molecules. If you get these names mixed up, again, it's not that big of a deal. I'm more focused this year on just understanding the process of the carbon recycling. All right, so I had to edit this whole big diagram to make it a little bit easier on you to recognize some of the molecules. And here I can show you what this original diagram looks like. It looks like this, all right? So what I did was, there's a lot of like, oh, we have like 3 of these, 3 of these, 3 of these. I said, you know what, that's a little bit difficult to visualize. So what I did was I actually copied and pasted and just made it what it looks like here. So we can see here, we have 3 of the RuBPs. We can see there's 15 carbon atoms here with the RuBPs. They're going to bond with these carbon atoms from CO2, and they're going to make that 6-carbon molecule. Don't memorize the name. That's going to break down to the 6 3-PGAs. We're going to use ATP and NADPH from the light reactions, and we're going to make 6 total G3Ps down here. Now, one of those G3Ps will go on to make glucose. The other 5 will be recycled to make the RuBP. Now, this process will continuously go around until we have enough G3P to make a glucose. So this whole process here turns twice because, again, we need 6 carbons for each glucose. We have 3 here. We need this to turn twice. This is confusing. You just got to memorize it. All right, this slide just reminds you again, you need 2 G3Ps to make 1 glucose. This whole process has to occur twice. That's it. Lastly, G3P is a very versatile molecule. We actually see it a little bit in our cellular pathways. G3P can make glucose, fatty acids, other amino acids, which then can go on. You can see this glucose phosphate makes fructose, makes sucrose, starches, cellulose. This is a very versatile molecule that the plant can use to make a bunch of different other polysaccharides.